have two of our very own today. So first, starting off is Mingzhu Hou. So Dr. Hou is a research scientist working in the functional neuroimaging of memory lab at CBL at UT Dallas. Her research interests include the behavioral and neural correlates of human memory, how they change with advancing age, and strategies to improve memory performance. So before joining CBL, she received her PhD degree in psychology from the University of Arizona. And for this presentation, Dr. Hou will be introducing her recent work employing fMRI and eye tracking to examine memory precision. So please help me in welcoming Dr. Mingzhu Hou.
For your introduction, Jessica, and hello everyone. Today I'm going to introduce some new findings from our lab in precision. So first, what is memory precision? Memory precision is the level of correspondence between the original experiments and the memory. It plays a very important role in our daily lives, help us differentiate similar information. So in a study conducted in the year of 2017, a group of participants were required to draw the iconic brand's logos based on their memory as accurately as possible. And here's a response from one participant drawing the logo of Starbucks. Based on the level of details within this drawing, you can imagine how confused this person will be when they see a similar coffee shop's logo like this. So in this case, the confusion as a result of the reduced memory precision will likely lead to a bad choice of coffee. And similar confusions also exist in other aspects in our lives. So now more and more behavioral studies are trying to understand the factors that could influence memory precision. And to our interest, it has been reported that memory precision can decline substantially with advancing age. And here's a result from a paper published 2 years ago. As you can see, memory precision declines from the young adult group to middle age and toward older age group. By contrast, retrieval success, which is a likelihood of retrieving a past event, regardless how detailed it can be, remains relatively stable with age. So in contrast to the accumulating evidence from the behavioral studies, very few studies have tried to understand the neural mechanisms underlying memory precision. So the primary goal of our research is to characterize the neural correlates of memory precision in young adults. Here are our memory tasks. Basically, we conducted 2 fMRI studies and recruited 2 overlapping groups of young participants. The 2 experiments share the same study phase, during which we presented unique object images to the participants. These images were placed on a random location along an invisible circle. Paired with each image, there was a small green circle. So participants' instructions were to move the cursor along the circle to the center of the image and try to remember both the image itself and its location. As a test phase of experiment 1, we presented both the studied and new images to the participants. And for each of them, the instruction were to move the cursor to the study location of the image. Participants can also take a guess if they cannot remember the location or they thought the image was fake. Then they perform an item recognition task by indicating whether this image was studied or new. As you can see, the location recall phase in this task, there was a temporal overlap between memory recall and the cursor adjustment. So to reduce the possible confound on the neural activities associated with these 2 processes, in experiment 2, we modified the test task by introducing a recall phase before the cursor adjustment phase. So in this phase, during recall, participants were required to remember the location in mind, and then to move the cursor along the circle to reflect or indicate the location. The other aspects of experiment 2 were the same as experiment 1. How do we measure memory precision with this memory task? Here we only focused on the studied images that were later correctly remembered. And for each of them, we computed the angular error or distance error between the study location and the location responded by the participant. Later, we just put all the distance errors from all the trials across all participants into a two-component mixture model. This gave rise to a uniform rectangular model reflecting the likelihood of lucky guess about location judgment, as well as a one-basis distribution, which reflected the likelihood of successful location information or successful location recollection with varying level of precision. So we further here, the kappa is the thread of the one-base distribution. It reflects the precision of the memory performance. A higher value of kappa is associated with a higher level of memory precision. So with this parameter, we further define location H trials as those who have more than 95% of probability falling in this one-basis distribution. For those trials falling outside the cutoff, we named or defined them as guess trials. Here are the model parameters derived from the two experiments. As you can see, performance was better for experiment 2 than experiment 1, reflected by higher kappa value and a lower guess rate. However, to ensure the comparability of the estimated activity across the two experiments, we chose to use the cutoff based on experiment 1. But I want to emphasize that when we use the experiment specific cutoff, our results are not changed. So in the following analysis, we include experiment as either a component, control, variable, or a factor, and we do not identify any significant interaction with the experiment. So how do we assess the effects of memory precision with this design? Here we took a categorical analysis approach. So within location H trials, we further chose two subgroups of trials that were associated with either high precision or low precision. And outside the cutoff, we choose a subgroup of guess trials. So we included a small gap within or between the adjacent categories to reduce the possible confusability between. Memory precision effect was evaluated by the difference between high and low precision trials. And to complement this analysis, we also conducted a linear mixed effects analysis to examine the relationship between trial-wise photo activity and the angular. By contrast, retrieval success effect was evaluated by the difference between low precision trial and guess trials. In our fMRI analysis, we choose to focus on two brain regions, the left angular gyrus and the hippocampus. Both of them belong to the so-called core recollection network, which is a list of brain regions that has been repeatedly reported to show sensitivity to successful retrieval. Although there were few studies trying to examine the neural correlates of memory precision, they all focused on these two regions. So in our experiment, we chose to use a combined AGLI, which is based on two peaks reported by the two fMRI studies on memory precision. For the hippocampus, we use anatomically defined hippocampus as the search space, and we employed an F contrast among the high precision, low precision, and guess trials. In that way, we were able to identify a cluster in the right hippocampus, demonstrating the main effect of memory type. So below, I'm going to introduce our findings about fMRI univariate findings from these two regions. So in left AG, we identified a significant memory precision effect reflected by the greater bold activity for the high precision trials and the low precision trials. This result was consistent with the results from the LME analysis, in which greater bold activity, the angular gyrus, was associated with a lower listener error and thus higher memory precision. In the hippocampus, we found similar results. First, we found a significant difference between the high versus low precision trials. And the high precision trials were also associated with greater goal activity than the guest trials. The LME analysis reveals that greater hippocampal activity was associated with higher memory precision. So our next step was to put the activity from the both regions into the same model. And with this analysis, we found that the AG and hippocampal activity independently predicted memory precision, suggesting these two regions might play unoverlapping roles in supporting memory precision. Our next question was that whether the neural activity in the two regions are sensitive or supportive of the recovery or reinstatement of the high precision information. So to address this question, we conducted a multivariate pattern similarity analysis. This was done in the anatomically defined angular gyrus and hippocampus. So in each of these regions, we extracted the trial-wise both activity from all the voxels belonging to that region, from both the study phase and the test phase. And for any given trial from the certain trial type, we computed the correlation between these trials' encoding pattern and retrieval pattern, as well as the mean correlation between these trials' encoding pattern and the retrieval patterns from all the other trials belonging to the same trial type. So item level reinstatement was defined as a difference between the within trial or within item correlation minus the between item correlation. With this approach, we were able to estimate the item level reinstatement for each trial type and for each participant. Later, we found that in the angular gyrus, the high precision trials were associated with above chance item level reinstatement effect. This was not the case for the low precision guest trials. And more importantly, the reinstatement effect was greater for high precision than low precision trials. Here you can see a small outlying data point. So after removing the outlying data, our results were not changed. So in contrast to the angular gyrus findings, we did not identify any robust reinstatement effect in the hippocampus. These are our fMRI findings. The second main question of our research was to examine whether the eye movements or eye fixations are sensitive to memory precision. So this question was motivated by a very well documented finding from studies using the so-called look at nothing paradigm. And here I have a small simplified example for this paradigm. In this example, at the study phase, participants learn different symbols, different colors, and placed at different locations. At test, the question was what's the color of the star? Even though no information about the location was asked, participants still tend to look at the quadrant associated with the star. So this pattern of results suggests eye fixations during retrieval might contain some location information of the. However, it was unclear whether the fixations are sensitive to the precision of the location information. So to answer this question, we acquired eye movement data during the second experiment. And we choose our time window starting from the onset of the image to the end of the recall phase. And to separate out or filter out any fixations that are related to the image instead of the location, we draw a square, four times the size of the image, and filter out all the fixations within the square. So fixation precision, the metric we want to use, is calculated as a trial-wise mean Euclidean distance between each fixation relative to the study location. This reflects how close people look at the location when they are trying to remember the study location. With this metric, we identify a significant effect of memory precision and retrieval success. From this figure, you can see the high precision trials were associated with higher fixation precision than the low precision trials and gas trials. And furthermore, there was a significant difference between low precision and gas trials. This effect was also confirmed by the LME analysis, where the greater fixation precision was associated with higher memory precision. We later assessed the view patterns of the fixations and whether they vary according to different trial types. Here I show you the plot in which we just normalize all the study locations and their associated fixations in reference to a single point. And the star here is a uniform study location across all participants and all trials. As you can see from the figure, for the location hit trials, the fixations were more centered around the study location. And this is more obvious for the high precision trials. By contrast, for the gas trials, the fixation patterns were more widely spread. So to quantify the fixation patterns, we computed a multivariate fixation pattern similarity metric. This was done by focusing on two trial types, location hits and gases. And for each trial type, we computed the mean correlation of the fixation maps across all trial types belonging to this trial type, as well as the mean correlation of the maps across all trials belonging to the two different trial types. So for any given trial type, the fixation similarity was computed as a difference between the within category correlation minus the between category correlation. Later analysis with this metric, we found that there was for both the high precision and low precision trials, there were robust fixation similarity effect. But for the gas trials, the similarity was at chance. Between trial types, between the high and low precision trial types, we found a significant difference. And the similarity for the high precision trials were also greater than the low precision and the gas trials. So this type of results suggests that people's view patterns become more unique as their memory precision goes up. So with all of these results, we can conclude that the hippocampus and the left angular gyrus plays a non-redundant role in the retrieval of high fidelity episode memories. Also, the angular gyrus might support the reinstatement of high precision information. And fixations during retrieval can track the fidelity of the retrieved mnemonic information, especially when it's related to location. So that's it. And I want to thank the whole FNIM lab, especially our RA team, for their support. And also, I want to thank our collaborators at the University of Arizona. This research is supported by NIH. And thank you all for your attention.
Right, great. We do have some time for questions. So does anyone have any questions?
First of all, wonderful. It's really exciting and very elegant analysis, and the results are very clear and exciting too. One thing you may have said, and I missed it, but how are you taking into account the subject level, the participant level in the analysis? You talked about the trials, but those are embedded in individuals that completed the task. Do you have ways of extracting individual level, like for individual differences between participants? Does it matter? Is everybody the same? Within individual, are there enough trials to make anything meaningful?
Mm-hmm. Yes. So in the linear mix analysis, effects analysis, we use the participant as a random intercept because we also try to use them as an randomly slope, but some models just run into the singularity error. But we did extract the subject level correlation coefficient from each type of analysis. And we found for the majority of participants, they did show the same pattern as we found from the LME analysis. So that suggests that these effects not only exist for the trial also for the subject level.
All right, I can ask a question. Why do you think, so looking at your bold data, why do you think that the low precision hits actually had the lowest neural activity compared to like guesses or the high? So this was your first experiment in your bold analysis. Sorry to make you go all the way back. Yeah, so why do you think it is, and why do you think that low precision was the lowest out of the three for the bold effects?
Yeah, that's a very good question. So in this task, as you can see, only asked information about location. So even though some trials might be categorized as guess trials, they might contain some recollection signal that is relevant to location. So I think the difference here, although it's not significant, different here for the location, low precision hits and guess trials might reflect that some guess trials still contain some episodic memory signal.
Okay, maybe another follow-up.
Yeah.
So you focused on the two regions, angular gyrus and hippocampus, of the recollection network, right? So the other regions within the recollection network, did you think are irrelevant for location, or just findings were not like more midline structured, midline parietal regions, for example?
So we did this our analysis based on prior research. These are the two main regions implicated in memory precision. And in addition to the ROI analysis, we also conducted a whole brain analysis, but we did not identify any significant precision. So for the retrieval success effect, we did find some clusters from experiment 1, but those effects are mainly related to the visual motor efforts, because in experiment 2, those effects were just almost gone. So, yeah. I mean, if we assess different features of memory precision, maybe other corrected regions will be involved. So.
Questions? Do we have any questions online?
We do not.
Great. All right, so we will go ahead and move on to our next speaker, but let's give a round of applause for Dr. Yin. So our next speaker is Dr. Qin Yin. Dr. Yin is a research fellow in the OFIN Lab of Cognitive and Brain Development at CBL at the University of Texas at Dallas. Dr. Yin received her PhD in psychology from Wayne State University in 2023 and is continuing her research here at UT Dallas. Her research focuses on the neural correlates of memory and memory development with a particular interest in developing memory representations. She uses intracranial EEG to investigate these processes, and today she will present her latest project on the development of the medial temporal lobe and its connection to memory development. So please help me welcome Dr. Qin Yin.
Thank you, Jessica, and hello, everyone. Today I'm going to talk about how the developing medial temporal lobe supports different types of memory representations and underlies memory development. So memory development isn't just about remembering more, it's also about what we remember. So one line of research has focused on the development of different types of memory representations. When we remember something, we might recall both the perceptual details, the specific features we saw or experienced, and the conceptual or abstract aspects of the event. So for example, if we look at this classroom picture, you might notice the red tables, the stuffed bear in the corner, the fish tank, and the green plants. These are the detailed surface level perceptual features. And on the conceptual level, however, you might simply recognize this as an indoor scene, or more specifically, a classroom, or perhaps even an elementary classroom. So studies examining memory development, including the well-known fuzzy trace theory, have shown that children tend to recall more of the exact details of an event. And as we move into adolescence, adolescents start to rely on the conceptual gist-based aspects of memory rather than just the surface details. And here I want to emphasize that for both types, I'm referring to how we access or rely on these memory representations during retrieval, not simply how much information is remembered. In fact, when measuring the amount of information recorded, adolescents often remember more details than children, but they also start to recall the conceptual aspects of past memories. And we know the medial temporal lobe is crucial for memory. A large body of research points to a functional gradient from the posterior to anterior of the medial temporal lobe. The posterior MTL is involved in processing contextual and spatial features. It's linked to returning memory details and supporting recollection of context and the details of an event. The anterior MTL, on the other hand, is more tuned to semantic and conceptual information. It helps us hold on to the gist of experiences and contributes to the sense of familiarity, even if we cannot recall every detail. So with that in mind, the central question we are asking here is whether and how the functional maturation of the MTL accounts for the developmental trajectories of memory representations we've just discussed. So to investigate this, we examined two key aspects of MTL function. First, we examined the temporal dynamics of activity in the posterior and anterior MTL to determine whether these regions specialize in processing perceptual features versus conceptual information during a thin memory task. And second, we analyzed encoding retrieval similarity to quantify the information that was reinstated when subjects recognize old pictures, specifically to examine whether these regions support different types of memory representation during successful retrieval. So today, I will present intracranial EEG evidence demonstrating the development of gist level or conceptual representations in the anterior MTL. And intracranial EEG recordings involve patients with medication-resistant focal epilepsy. As part of their clinical care, these patients have electrodes implanted to pinpoint the area causing seizures. So on the slide, you can see an example of electrode implantation during surgery, a CT scan, and a demonstration of electrode coverage. And after surgery, patients stay in the hospital for monitoring. It's during this time, we invite them to participate in our cognitive tasks. And the major advantage of IEG is that we can get signals from the ventral regions, like the MTL cortex. And it can also capture brain signals with both high spatial and temporal resolution. And using these recordings, we can get a much more detailed look at the dynamics of MTL activity. Here is the electrode coverage from our 23 subjects with electrodes placed in the medial temporal lobe cortex. And for our analysis, we looked at the signals separately in posterior and anterior MTL. And we divided the MTL into posterior and anterior using the midpoint between the most posterior and most anterior electrodes across subjects. So while these subjects were undergoing the IEG monitoring for failure management, we asked them to perform a thin subsequent memory task. So during the study block of the task, subjects were presented with pictures of scenes and asked to indicate whether each scene was an indoor or outdoor, which is a conceptual level decision. And in the test block, old pictures were intermixed with new ones, and subjects responded old or new to indicate whether they had seen the picture before or not. Old pictures in the test block were labeled as hit or miss, and the corresponding pictures from the study block were labeled as subsequent hit or subsequent miss, depending on whether they were remembered or forgotten in the test block. So I want to emphasize here that our task doesn't require subjects to focus specifically on either details or higher level features. So it gives subjects the flexibility to remember or recall whichever aspects of the scene they naturally prefer. So this allows us to observe individual or developmental differences in the types of memory representations on the brain level if they are any. And we first examined the temporal dynamics of brain activity relative to scene onsets and to subjects' response time on each trial. So here we looked at high frequency activity between 70 to 150 Hertz. Activity within this frequency range reflects local neuronal activity with millisecond temporal resolution. And here I'm showing you the high frequency activity from one study trial recorded from a posterior and an anterior MTL electrode. And in these examples, you can see how the activity in the posterior MTL increased after the scene onset, and how the activity in the anterior MTL increased around the response time. So here I'm showing you just one trial example, and to look at the activity relative to both scene onset and also each trial's response time on the group level, we put all the trials across subjects together and sorted them by response time separately for subsequent hit and miss trials. And in this plot, the y-axis represents individual trials ordered from shortest to longest response time. And the x-axis shows time relative to scene onset. The white vertical line indicates scene onset, and the black line marks the response time of each trial. The yellow color indicates increased activity. From this plot, we can see that the posterior MTL exhibits increased activity shortly after scene onset. And the activity in this region is not related to scene category, the indoor-outdoor decisions. In contrast, the anterior MTL does not show increased high-frequency activity, after scene onset. Instead, activity in this region increases around the response time for each trial. So for the activity differences analysis between subsequent hit and subsequent miss trials, we focused on the first second after scene onset in posterior temporal lobe and aligned the data to each trial's response time in the anterior MTL. So here I'm showing you the averaged activity on the group level for subsequent hit and subsequent miss trials. We can see that in the posterior MTL, again, increased activity after scene onset, but higher activity in subsequent hit than miss trials. Here, the gray shaded areas indicate significant difference between subsequent hit and miss trials. And in the anterior MTL, after aligning the trials to subjects' indoor-outdoor response times, we observed activity differences between hit and miss trials before response. But we note that here the difference is mostly due to the decreased activity in subsequent miss trials. And I want to point your attention to the increased activity around the indoor-outdoor judgments for both subsequent hit and miss trials. And these findings suggest that The posterior MTL is involved in scene perception, while the anterior MTL is more engaged in conceptual level scene categorization. So here is the data for then what about retrieval? And we took the same approach to examine the activity during retrieval. And in our task here, again, is recognizing previously presented scenes. Again, we stack all the trials across subjects and sort them based on their response time. Similar to what we saw in encoding data, posterior MTL activity increased shortly after scene onset. And activity in this region is not related to memory decision. Here, the memory decision is indicated whether it's an old picture or a new picture. And in the anterior MTL, activity is not related to scene onset, but instead around each trial's memory response time, again, old or new, we have the activity increase around this memory decision time window, especially during HIT trials. So for the activity difference analysis between HIT and MIS trials, we took the first second after scene onset in the posterior medial temporal lobe and aligned the data to each trial's response time in the anterior MTL. Looking at the group level average activity, we can say that in the posterior MTL, activity increased after scene onset, but there are no differences between HIT and MIS trials. And in the anterior MTL, we're aligning the data to response time. Activity increased around memory response. But before the old new memory decision, there are also no activity differences between HIT and MIS trials. So the difference between HIT and MIS occurs after a memory decision. And we think the increased activity in HIT trials after the old response may suggest the role of anterior MTL in post-decision evaluation and verification. But here comes the question. In these regions, there are no activity differences between HIT and MIS for old-new memory decision. And what makes subjects recognize the presented as an old one? So this brings us to our second aspect of MTL function, the encoding retrieval similarity or memory reinstatement. Previous studies demonstrated that memory retrieval depends on reinstatement of encoding-related activity patterns, typically measured as encoding retrieval similarity. So with a high temporal resolution of intracranial EEG, we can compute encoding retrieval time similarity matrix for each brain region. This matrix quantifies at each time point of retrieval how similar the brain activity is to specific time points during encoding. It measures during retrieval when the brain reinstates activity patterns of encoding. So previous IEEG research has revealed two reinstatement patterns or shapes in the encoding retrieval time similarity map. The first pattern happens after cue or stimulus onset during retrieval. It shows a diagonal shape, particularly towards the upper diagonal. And this indicates that retrieval recapitulates the sequential perceptual features of an event, but at a faster time scale. And the second pattern is observed around the time of retrieval response, such as during recall. And in this case, reinstatement forms an expanded rectangular shape in the time similarity map. This suggests that around retrieval decision, subjects are likely retrieving the entire memory event or activating a higher level conceptual representation. Therefore, the retrieval activity during this response period recapitulates a range of encoding moments. creating a reinstatement pattern that spans an extensive encoding time window. So in our data, we analyzed encoding retrieval similarity using the power spanning the full frequency range from 1.5 to 250 Hertz. And we think that power from the full frequency range would best capture the overall brain status during each time point of encoding and retrieval. On the left, you can see the plot of power across the full frequency range for encoding and retrieval. And on the right is the encoding retrieval time similarity map. Each point in this matrix, again, represents a correlation between the power at a specific encoding time and a specific retrieval time. For example, the data highlighted in right circle is the correlation between retrieval at 1 second and encoding at 0.5 seconds. It gives us how similar the brain activity during retrieval at 1 second is to the activity during encoding at 0.5 seconds. So in this similarity map, the white solid lines mark the encoding and the retrieval stimulus onset. The black dashed lines indicate the averaged encoding and retrieval response times. And the white dashed line is the diagonal, marks the equal time points between encoding and retrieval. So we can see that in HIT trials, there is high similarity shortly after stimulus onset, and the similarity follows the diagonal, indicating the activity patterns during retrieval closely mirrors the temporal sequence of encoding. However, in the similarity map for these trials, not only is the similarity much weaker, it also appears predominantly below the diagonal. And here I'm showing the differences between HIT and MIS. The differences map gives the memory reinstatement, which is most prominent above the diagonal line. And this memory reinstatement pattern suggests that posterior MTL recapitulates the sequential perceptual features of the memory trace at a compressed timescale during successful memory retrieval. So what about anterior MTL? And when we examined high-frequency activity in this region, we observed a response-related activity profile reflecting either higher levels in conceptual representations during encoding or memory decision process during retrieval. So to check whether memory reinstatement in this region is also in anterior MTL is also tied to response, we first did the encoding retrieval similarity analysis aligned to stimulus onset, exactly as what we did for posterior MTL. And for comparison, the similarity map for the posterior MTL is shown again on the right. As you can see, especially for HIT trials, the similarity in anterior MTL does not follow the diagonal pattern observed in the posterior MTL. Instead, it displays a more rectangular shape and closely aligned with the average retrieval response time. So to accurately capture the response-related reinstatement in anterior MTL, we conducted the similarity analysis by aligning the data to the response times. And in the figures, we aligned the analysis to participants' encoding and retrieval response times. The black lines mark the response times for encoding and retrieval. And in the time similarity map, the white dashed diagonal line marks the same time points between encoding and retrieval relative to subject's response. And notably, the similarity does not follow the diagonal. And in HIT trials, we observed high encoding retrieval similarity before both the retrieval response and encoding response, forming a rectangular pattern. And in contrast, the similarity map for mixed trials show much weaker similarity with no clear pattern. And the difference between hit and mixed trials captures the memory ring statement. And we can say that reinstatement predominantly occurs before the retrieval response time and is divided into two clusters by encoding response time. So if you can recall in anterior MTL, before response, there is no activity difference between hit and miss, but there similarity difference between response. So it's the reinstatement in this region helps subjects to recognize the picture is an old picture. And in this map, these clusters likely suggest that in anterior MTL, successful memory retrieval involves reactivation of higher level conceptual representations before old new memory response. So having identified distinct patterns of memory reinstatement in these regions, we can now answer the question we asked at the beginning of the talk. Are there any developmental differences on these two types of memory representations? So using the memory reinstatement cluster observed on the group level, we extracted the individual reinstatement measures and examined age-related differences. And we found that the reinstatement in the posterior MTL was not associated with age. In contrast, in the anterior MTL, reinstatement increased with age, suggesting that conceptual representations in this region develop with age. This might support the conceptual or gist level memory retrieval in adolescence. And to summarize, first we found distinct temporal dynamics in the posterior and anterior MTL. And this dynamics suggests that the posterior MTL supports scene processing and perceptual representations, while the anterior MTL supports conceptual representations. And developmentally, there is no age differences in conceptual representations in the posterior MTL, but conceptual representations in the anterior MTL develops with age. That's all for my presentation, and I'd like to thank my co-authors, lab members from the lab, and families and patients who participated in this study. And thank you all, and I'm happy to take questions.
Great. Do we have any questions?
Yeah, I did. I also thought you did. So I think I asked you this. Can you go back to one of the last slides where you were showing hip miss contrast? Sorry, I didn't look back again. This. What is that? What do you think about that low frequency pre-onset delta band? It's just a group average, right? That we're seeing at the bottom left. Yeah, what do you think about what is that?
You mean this?
Yeah.
Slow thing.
I think we can look at this compare with HIT trials. So we don't have this in HIT trials, and I think within this range, it's more about theta or like slow theta. So if we interpret it from the low frequency power desynchronization, it may suggest that this region is like no formation processing in this region during unsuccessful retrieval, or we can simply interpret it as the subjects are not paying attention to the pictures during both encoding and retrieval.
reliably at the group level, right?
Right.
Yeah,
Right, group level.
but that's cool. Thank you.
Yeah, thank you.
Also said that there's like, because it's MTL and not hippocampus, there's a lot of effects that are kind of flipped.
Okay.
That we do have a negative, like we do hit versus miss. That comes as negative because it's higher.
Yes.
Okay.
What relation between age and the anterior MTL effects?
Yeah.
To what extent does that relate to performance? Do you know?
So that's a great question. And in our task here, as I introduced, we just ask them to indicate if it's indoor, outdoor during encoding, and during retrieval, just to indicate if it's a new or old picture. So we don't have performance measure on which representations the subjects are using. And I think this just gives us a grade level estimation of when subjects recognize the picture as an older one. Maybe in the older subjects, adolescents, they also have this higher level of reinstatements.
Follow up on this, and if you'd include performance in the model, would it change? I think the question is like performance between individuals.
So in this region, the restatement in the anterior MTL is not related to the recognition accuracy.
Okay. So for that particular performance measure,
Right.
maybe it might be related to the measure if you.
Yeah, so if we have like different performance measures to measure if they recall more of the details or more of the abstract, this might be a way we can correlate the restatement to that performance measure, but unfortunately in our task we don't have that. Thank you.
Other questions?
I have a quick one. For your delineation of the midline between like posterior and anterior MTL, did you do that per hemisphere or was it like one midline?
It's across both hemispheres to have a most anterior boundary and a most posterior boundary, and use that midline for both left and right hemisphere to divide this into anterior versus posterior.
Anything else? All right, great. Well, let's give another round of applause